messages. For the wire, just be careful. Be careful of the wire. 1, 2, 1, 2, 3, 4. I think that is working. I think that is working. Yes. Ooh. But that's not very good, is it? The wrong video. hand those out for me, please? Could you please hand those out for me, please? Yeah, chuck those down there. That'd be great. Thank you very much. Can you please sign that and pass it around? Could you please pass those out for me, please? Could you please pass those out for me, please? Could you please pass those out for me? Just one each, thanks. Boys and girls, ladies and gentlemen, fantastic to have you here. Not bad, 7 minutes late. I was fearing it was going to be longer than that. There's so much bits and bobs that have to be set up beforehand. I had to bring down some brains. I had to set this up. So this is being recorded, it's being streamed live on YouTube at the moment, so please do not swear or otherwise impeach yourselves. I tried to record all of the lectures on YouTube, and I got quite into the habit of doing that. And then last night, I just read that Google Hangouts has been scrapped, so I had to rediscover how to record live on YouTube without Google Hangouts. So this is the first time I've done that. So I hope this works. It seems to be working at the moment. But hopefully if things go well, all the lectures will again be recorded, that I do anyway, will be recorded on YouTube. I can't ask the other lecturers to do that, but everything that happens in here, when this red light is on, it means it's getting recorded automatically in terms of the slides and the audio onto Blackboard anyway. So in fact there's 2 recordings going on at the moment. The other thing is that there is a register that is being passed around that you need to sign, so we know that you're here. I'm afraid the fact that it's live streamed and recorded on YouTube doesn't mean that you don't have to be here in person, but I'm sure you wouldn't want to miss out on the face-to-face excitement, spontaneity and pleasure. And also you should all have one sheet for a colouring exercise in a minute. I do enjoy colouring in myself, I find it very relaxing. Everyone should have one of these. There you are. Okay, so this unit is called Brain Mind Education. I should also say a couple of people wanted to talk to me while I was setting up, but I couldn't talk to them. But if they want to catch me at the end, we're hoping to end at around about 1:40. All right, that's about 2 hours and 40 minutes time, but it will be a 15-minute break in the middle. I think there's some seats over there, guys. It might be easier, it might be quicker actually for you to go back in the other door. It's called Brain-Minded Education. It's about empowering you with the tools to be able to approach the neuroscience literature for topics that are of interest to you and be able to understand that literature in a critical manner. Okay, that means being able to understand the limits of what you're reading as well as the insights that it may provide. If you're going to read anything, the most important thing you should be reading are e-journals, papers, research papers that you can find using the web of knowledge, or sometimes known as the web of science. Do you know what I'm talking about? Put your hand up if you know what I'm talking about when I say web of science or web of knowledge. Right, that's not enough. You all need to find out about the web of knowledge or the web of science. It gets called both things, which I find very confusing. You can use Google Scholar, but actually you know that something on the web of science, web of knowledge, is peer-reviewed. It's also very easy to download onto your citation software. That's something else you might want to think of, even this early in the year. Think about using citation software for your essays. Okay, it's just a little hint. The most popular, I think, is called EndNote. I believe that you can use it either for free or very cheaply via the university, but do find out about it. It means that when you find a paper on the web of science or web of knowledge, you can just click a couple of buttons and it automatically gets loaded into your private library. And then when you want to cite that as evidence in your essay, you just press another button and it goes Howard Jones et al. 2015, and then all the details get automatically put at the end of your essay. It's fantastic. And then when you find out that in fact you've used the wrong format for your essay, you can just click one button and they all get automatically put into the correct format, Harvard, APA, whatever it is you wanted. So it's a really fantastic way of ordering your life and making your essays easier. And then it comes to your dissertation, and by that stage you'll already have loads of papers in your private library that you'll know roughly something about, so you'll be able to draw on them easily in your essays. Okay, enough about that. Brain, Mind and Education. So if you're going to buy a book, however, I would recommend my book. Actually, I can see that there's a problem here because there's a microphone here, and that may not have been picking me up. I think it's got so much technology. Something else I haven't switched on. I can't even open the damn thing. I think I need to have this on as well in order for it to record on the university one. Hello, hello, hello. Hello, hello, hello. Can you hear me? Is anything coming through? Is it coming through? Are you sure? Hello, hello, hello. I don't know about that, actually. I'm not confident. Hello. Ah. That's more like it. Otherwise it would never have got recorded on the university one. Also, you can hear me better now, can't you? So yes, of course, I would recommend my book because it tells you something about how neuroscience connects to education. I think we're all interested in education, aren't we? Yes, good. If you're going to buy a book which is focused just on cognitive neuroscience, I would really recommend the Jamie Ward book, okay, because that is it's a student's guide and it deals very broadly with almost all of the subjects in cognitive neuroscience, in a very introductory manner, which is really helpful. And there are also online resources available for that book and CD-ROMs and things, so definitely I would recommend that book. Right. Timetabling issues, some slight complications. For those of you who are in my wonderful introduction when I had all these errors on the slide, I think they're corrected now. But please note that this lecture and the next lecture and also the lecture after half term. Sorry, I'm going to go back to something a lot more. See, there's some bits of technology that you can't rely on, really. So these two lectures are 3 hours long, and then the lecture after half term is also 3 hours long. Okay, this is because this term I'm going to be based in Geneva at UNESCO doing a fellowship there. But I'm commuting back to do these lectures. It does mean communication will sometimes be a little bit more difficult than usual, but hopefully it shouldn't present any problems. But it does mean that the most material outcome is that these 3 lectures are 3 hours long, so please come with prepared mentally for that. I've tried to make them interesting, so it shouldn't be too bad. There is an optional workshop. You will be allocated to one of 2 groups in that half term. That will be about choosing a topic and how to write scientifically in your assignment. You don't have to attend it, but I do strongly recommend that you do. There is a multiple choice exam on the 7th of November. It's going to be based on Neurons and Neuroanatomy, which is just about what we're going to do now. After the break, we'll be doing development. It will not include development, okay? It will not include development. Next week, we're going to be doing learning and memory. It will include learning and memory. All right, so I'll be clear about what it's going to include. If at any point you're not clear, when I'm presenting the slides, you can put your hand up and say, will this be in the test? And I can tell you whether it might be. If it's not on the slides, and I mean not on the slides for neurons, neuroanatomy, learning and memory, you can't be tested on it, okay? So I'm not allowed to do that. I wouldn't do that to you. And there is also a revision session on the 17th of October with Dr. Katie Blakemore, which will be fun, because you'll be playing a game during that session to help you remember. Neuroscience and Education Pathway. People on the Neuroscience and Education Pathway, could you please identify yourself? Small but elite group. I'm joking, of course. Everyone's very important. These people, we're supposed to have a little social meeting together at 2 o'clock today. It is going to be down at Bristol, because we didn't really organise the trip very well in Welcome Week, so we're running it again. So we're going to meet at 1:45, so the lecture's got to have finished by then, outside the graduate school here, and we'll walk down to at Bristol and we get a free ticket in. Any questions about that before I go on to the more interesting stuff? This is making me feel very confident. No? Okay. Neurons and neuroanatomy. These are the things that we're going to be doing. Most important thing, I suppose, to look at straight away is that I will not be testing you on which Brodmann area is which. Right? There's about 45 different no, more than that, 48, 50? Under 50 Brodmann areas in your head with numbers on. I'm not in your brain. I'm not going to be testing you on which one is which. Okay, let's make that clear. The neuron. Who's done GCSE biology or any biology at all ever in their life? Well, you probably know all about the neuron then. That's the nucleus. These are the dendrites. An action potential, a little piece of electricity, an electric charge, is actually sent down the axon onto these presynaptic terminals. That is the communication of information. So this is the cell that does all the information processing, or most of it, in your central nervous system. It's a very important cell, therefore. It evolved specifically to do information processing. Around this axon, which can actually be very long, they can be as long as that. You know, they can be stretching down your leg. But in order to insulate it, we have myelin. Myelin is white, and it actually is what gives rise to the white matter in your brain. The grey matter is because all the neurons are sticking together densely. The white matter is due to the insulation around the axons. So we have grey matter and white matter in the brain. Generally, we tend to be more interested in the grey matter, but actually the white matter can be interesting too. You can see there's a Schwann cell here that's wrapped itself around, making myelin, trying to keep that axon insulated. If we didn't have myelin, then the electric charge would leak out. And it would leak out to other axons, and we get a mess in terms of all the information that was going on in your brain. So myelin is very important. And we have the nucleus here. We also have nodes of Ranvier which are not myelinated. So it tends to sort of jump across those nodes of Ranvier. But most of it is insulated, so that tends to help carry the charge efficiently from the axon down to the presynaptic terminals, where it may connect to another neuron. If it does connect to another neuron, it will be going on to the dendrites here of it. Now, how does this signal get sent? Well, to understand how this electrical signal gets sent, you need to think about the distribution of the ions. An ion is a charged atom or molecule. Okay, it's got a charge on it. And the distribution of those ions brings about a difference between the amount of charge inside the cell relative to what is in the outside world. So this is what I just showed you there. We've got the inside of the cell here, there's the nucleus, and we've got the outside world here. And I've just put it into a sort of a diagrammatic form here, so the purple is still inside. And you can see that we have protein molecules here, slightly negatively charged, potassium ions, which are positively charged, chlorine ions, sodium ions. We also have them on the outside as well, on the outside of the cell. But this membrane has certain properties. And at rest, the potassium ions cannot cross it very easily. The chloride ions, sorry, I beg my pardon, the potassium ions can cross it very easily. So the potassium ions are sort of leaving, if you like, quite easily. Now that's positive charge leaving, okay? We have chlorine ions and sodium ions that have a bit more difficulty in crossing it. We also have charged protein molecules carrying a negative charge that cannot cross it, so they're staying inside. And we have a pump, which is pumping out 3 positively charged sodium ions for every 2 positively charged potassium ions coming in. Now all of that adds up to a negative potential for the inside of the cell. So relative to the rest of the world, that cell is negatively charged. It has a resting potential of minus 70 millivolts. Something has to change the polarity of that cell a little bit. Well, for example, it might be some peripheral neuron, maybe the neuron at the back of your eye, a retinal neuron, that responds to light. Okay, so those sorts of peripheral neurons, they sort of transduce the energy from one form into another. So they take the light energy, turn it into a little bit of electricity. That connects to a neuron that then has its electric potential raised slightly to this threshold. Once that happens, a whole cascade of events occurs. Once it's crossed this minus 55 millivolt threshold, you start getting the sodium channels, which are on the membrane of the cell, opening, which then allows the sodium ions to come in. And then we have the potassium, so that means we're getting depolarization, because a positive charge is coming in. That means it's becoming more and more positive. And the electric charge of that cell is going up and up and up. It's depolarizing. When it gets to that point there, the potassium ions can now go through their channels, which means that it can start going negative again. So the potassium ions are now sort of leaving, if you like. It's getting depolarized again. Repolarized. It's coming back down to its negative point. It swings down much further than its original resting potential. Then it comes up to its resting state. And that happens automatically. It happens automatically once you raise the potential of the cell by about 15 millivolts up to that threshold. I think this is supposed to... Oh, it does. That's very good. So this tells you what happens after that point. Because basically, that repolarization and depolarization occurs all the way down the axon. And so what would normally be negative momentarily becomes positive. And so you get a transmission of the electric charge down the axon. And as I say, these things can be like about a meter long, these axons. These cells very, very small, but these nerves are actually very long. So it's a way of transmitting an electric charge through the body. Although most of the axons in your brain, of course, are very quite small, a few centimeters or less. So we now have a means by which we can communicate information. A piece of electricity here can end up... This thing doesn't work, actually. A piece of electricity here can end up down there. And if we have neurons that are joined together, then we can see electric charge actually travelling through that neural network. So we may have an input here. Again, it may be from some peripheral neurons. It may be for touch. It may be pain or whatever. That original input from the outside world enters your neural network and it travels through the neural network. And you get an output. You get an output here at these three sets of presynaptic terminals. Now this means that we have the potential to transform information, because whatever was coded here ends up coded in a different way there. There is a mapping of one set of information there to an output set of information there. So, for example, this could have been sensory input. It could have been about something visual, and this could be about motor output. It could be about moving your hand or something. So in a very simple way, you could end up with a neural network with these 6 neurons processing information from one form, visual, an input into an output in terms of actually action. And in fact, most neural circuitry that's evolved, that's what it's about. It's about transforming sensory input into action output. That's how we developed our nervous system. Although we're going back here to jellyfish. Jellyfish are probably the supreme processors of sensory information. They are the most efficient swimmers in the ocean because they have neural networks that go around the perimeter of the jellyfish. And that means they're able to sense what's happening in the water and what's happening with their bodies and turn it into action very efficiently. The wonderful thing about jellyfish is that they have neural networks that allow them to be incredibly efficient movers. And they can also defend themselves in certain situations very effectively as well. But they actually have no brain. So they can learn a little bit, but they're not very intelligent. Nevertheless, they do have neurons. So that's how our neurons evolved. Started off probably in jellyfish. So we should thank the jellyfish for our neurons. So electric signals travel in a complex, interconnected manner. The question is, what defines how an input signal here becomes an output signal there? Well, what actually defines it is the efficiency of these connections here. Because depending on how efficient they are, that is going to determine how much of the electric signal going in actually gets transferred to the next neuron. So the efficiency of these connections, if you change the weightings, the efficiency of those connections, that will change the way in which this input gets transformed into that output. And that's basically how we think. That's the basis of our thinking. Okay. I suppose the next question is, well, that's the basis of our thinking. What is the basis of our learning? What would have to change in this in order for us to learn? Okay, so we've got different weightings here and these connections that are transforming this information into that information. But supposing we wanted to change the way we think, that's called learning, basically. If we want to change the way we think, what do we have to change? The connections, the efficiency of the connections. Okay, so that's called learning. Oop. Right, so we have information superhighways which are made up of axons that are myelinated, insulation. You will come across 2 words which are important. Afferent, which means axons going to something. So if I say that this is, I'm talking about the hippocampus, and I say this is an afferent axon, then it means it's arriving at the hippocampus. If I say it's an efferent axon, then it's actually leading out. It's the output of the hippocampus. The myelin insulates, speeding up the signal between the nodes of Ranvier. Any loss of myelin, and that can happen with viral infections, it also happens with multiple sclerosis, can bring about muscle weakness, seizures. And myelin is white, hence we get white matter in the brain. Neurons are grey matter. And where you have a very densely packed set of neurons, we call it a nucleus. Okay, it's a bit confusing because you've got a nucleus in a cell, haven't you? But this is a different type of nucleus at a different scale. So we talk about the nucleus accumbens in the brain, and then you know that that's a very dense region of neurons that are packed together. Now, I don't want to kid you that neurons are the only the only cells in the brain. It's not the only cell in town. There are many other cells which are very important. I'm just going to go briefly through some of them. You have cells such as glial cells, which are the housekeepers of the central nervous system. There's an awful lot of neurotransmitters that are produced, and which are excess, and there's a lot of rubbish produced that you have to clean up all the time. And glial cells are very important for that. We also now believe they have some role in cognition as well, interestingly. Macroglia are the big glial cells. They remove, for example, excess potassium. You're probably wondering, in the previous example, what happens to all that potassium that is sort of coming in and out of the cell? Well, astrocytes are handy for cleaning that up. We also have two types of macroglia, bigger types of glial cells, that make myelin. Oligodendrocytes make myelin, and they are a cell which can actually wrap around several axons at once. Schwann cells also do a similar thing, but they can only wrap around single cells, and they're only found in the peripheral nervous system. The peripheral nervous system is basically everything other than your brain and your spinal cord. Then we have little glial cells, which are very good at cleaning up debris, and they multiply with damage. So there's a massive increase in the number of microglia when people have a stroke, because suddenly the microglia get into action and they're trying to clean up the debris that's been created. Now, I did mention what is the central. I mentioned the central nervous system and the peripheral nervous system. So let's just very briefly talk about this. Now, we're beginning to talk about the divisions in terms of neuroanatomy, and this is pretty much the highest level of division. How can we divide up the nervous system in a way that we can talk about it easily? Well, the first thing that people do is to say there's a peripheral nervous system and a central nervous system. The central nervous system is the brain and the spinal cord. All the rest of the nervous system is the peripheral, and that's divided into two parts. Remember, I talked about neurons being particularly evolved to process sensory input in and action input out. Well, that's your somatic system, and it's part of the peripheral nervous system. Jellyfish do that very well, but they don't have a brain. They don't have a central nervous system. The other part of our peripheral nervous system is the autonomic nervous system, which is all about controlling your internal organs and your glands. It's responsible for your fight and flight system. So when you have to increase energy, then your sympathetic nervous system is going to get into gear. But when you're resting and digesting, which is also very important, you're reducing energy, we call that part of the nervous system the parasympathetic nervous system. Okay, we're coming up to a little bit of fun soon. Now you're going to have to start orienting yourself. I'm afraid there's all sorts of very strange terms that brain scientists use, and you will come across these being used very casually in neuroscience papers. And there's no escaping trying to learn what they are. So this is an important part of your test. You will definitely be tested on this. The first thing you have to remember is that I've put some of the words which mean the same thing together. That's because we're only dealing with the human brain. If we were dealing with animal brains, you would find that dorsal and superior are not the same thing. But I don't think you're going to be looking at rat brains or many other brains. So we're going to deal with these terms as if they mean exactly the same thing, because in the human brain, they do. We have dorsal superior. Okay, that's the top of your head, top of your brain. We have ventral inferior, that is towards the bottom of your brain. We have rostral anterior, towards the front. And either of those words could be used. So you might look at something and they'll say, in the rostral regions of the frontal cortex. And that means at the front of the frontal cortex. Or they might say in the anterior regions of the frontal cortex. It means exactly the same thing. They do it just to catch you out. And then at the back we have the caudal and posterior regions, travelling towards the back of the brain. By the way, we know this is the back of the brain. The easiest way of looking at that is, well, you've got the cerebellum here, which is sticking out, which is a little cabbage part of the brain which sticks out underneath. It's the part of the hindbrain. And I guess the other thing is this here, which tells you that there's the temporal lobe here. It's sort of like a flap. It looks a bit like a flap. All right. So you know that this is the front, and this, because it's the cerebellum hindbrain at the back. I sometimes take these things for granted, and I forget that you may not have looked at a picture of a brain before. It gets worse. These are other terms that you're going to come across. Ipsilateral. We had ipsilateral activation in the frontal cortex. That means it's on the same side. Bilateral, so it was on the same side, so it could be here and it could be there, for example. It was on the left, ipsilateral left regions of the brain. If it was bilateral, then in fact it's on both regions of the brain. I'm going to be testing this on this in a minute, actually. Okay, so yes! Yeah, I noticed somebody suddenly sit up then. That's great. Okay, so bilateral. Contralateral means opposite. Okay, so, you know, it may not be exactly balanced, but you might say contralateral activation of the motor cortex to moving the right hand. Well, in fact, that is going to happen because the right hand is actually related to left hemisphere motor cortex. So when you move the right hand, you should see contralateral activation in the left hemisphere. Medial means towards the middle. Lateral means away from the middle. Proximal means near, distal means far. I think those are probably the most annoying phrases, because why don't they just say near and far? I don't know. That's no excuse, but you will come across them. Proximal, distal. And then you're also going to see brain images. And that is kind of like often a slice through the brain. But which slice? Is it a slice like that, a slice like that, a slice like that? If it's sagittal, it basically means down the middle. What's a good way of remembering that, I wonder? Any ideas? Sagittal. Yes, okay, so Sagittarius is the archer. That's good. And they've got their bow like that. See? Okay, Sagittarius. Very good. Horizontal, well, that's pretty easy, except that sometimes they call it transverse. So you might see a transverse section, and it basically is a slice going through like that, or a horizontal section. Sagittal, transverse. And then the last one is coronal. That's between the ears, like that, coronal section. So much Latin. So much Latin. Right, okay, so Simon says, for people who are not accustomed to our nursery rhymes, we have a sort of not a nursery rhyme, a nursery game, really, with small children, where we say, Simon says, rub your tummy, and everyone has to rub their tummy. Okay, and Simon says, clap their hands. Well, this Simon is a neuroanatomist. Okay, so Simon says, dorsal superior. Rub your dorsal superior. Very good. Very good. Okay. Simon says ventral inferior. That's it. That's very good. That's very good. That's very good. Simon says rostral anterior. Oh, yes. Very, very good. Very good. Yeah, yeah. Simon says caudal posterior. Oh, very good. Very good. Yes. Simon says sagittal. Yay. Yeah, yeah. Very good. Simon says transverse. Woo, yes, very good, very good. Simon says coronal. Yes, yes, that's very good. Good, we're getting it, we're getting it. Okay. Oh, I've just done that one. Simon says, use your hands to indicate contralateral. Oh, well, it's sort of, yeah. Move the right hand and then left part of the brain is activating. That's contralateral. Or the other way around. Like that, yeah. Use your hands to indicate distal. Far away. Use your hands to, Simon says proximal. Okay. Simon says bilateral. Excellent. Both sides of the brain. Right. Good. Excellent. Oh. I know it's hard. But actually, you've already done this. So it's not so bad, really, because you've already just done that. And you've done this. Because we said we had the central nervous system, peripheral nervous system. We talked about how the peripheral was divided into autonomic and somatic, and the autonomic is also divided into sympathetic and parasympathetic. So that's all done. We said the central nervous system was in terms of the brain and the spinal cord, and this is obviously the bit that we're most interested in, isn't it? Oh, we love the brain. So let's divide it up further. And we divide it up further into three parts: the forebrain, the midbrain, and the hindbrain. And the midbrain is divided up into the mesencephalon, which becomes the tectum and the tegmentum. The hindbrain becomes the metencephalon, the myelencephalon. This becomes the pons, the cerebellum, and the medulla, and you haven't got a clue what I'm talking about, but let's move on and have a look, shall we? Whoop. Important thing about, well, one interesting thing about the three parts of the brain is the way in which the forebrain looks massive compared with other animals. So this is a shark, and you can see they've coloured the different parts, the different brain regions, with the same colours for the human and for the shark. And you can see that this telencephalon, which is part of the forebrain, is absolutely massive. You wouldn't even know the rest of it existed inside the human. You can hardly see it. Whereas in the shark, it's really quite moderate. And this has given rise to the idea that humans are particularly intelligent because we have very big forebrains. But actually, I hate to tell you that the most important thing about us is that we are big primates. We have exactly the brain that you would expect for a primate with a brain of our size. Okay, because actually as the brain scales up, so the forebrain naturally gets bigger. It's one of the things that they've found with primates. So that's a bit of a downer, isn't it? We're all basically just big primates. So this is how you might see the hindbrain, the midbrain, the forebrain. This is a sagittal section. You already know what I'm talking about here. It's a cutaway like that. And we can see the forebrain here. That's very important. We see the midbrain, titchy little midbrain here. It's very small, really. And then we have the hindbrain, where we've at least got the cerebellum contributing to that. So, we can see it poking out. While we're on the subject, can I just point out the cingulate cortex here, which is an island of cortex inside the cortex, if you like. Because the cortex, the most important thing about the cortex is the surface. And one way in which you can increase the surface is to scrunch it all up. So in order to get our larger amount of cortex in, it's all become quite scrunched up. That's why it's all wrinkly. If you unscrunch it, it becomes the size of 2 tabloid newspaper pages. But in the scrunching, you end up with islands inside. And the biggest island is the cingulate cortex here, which you can see when you do a sagittal section. And it's an important region of the cortex because it's very involved with paying attention. Over here, I just wanted to show you the brain 6 weeks after conception. You can see that it's already divided itself into these different regions. So we've got the telencephalon here and the diencephalon. This is the forebrain. We've got the mesencephalon, which is the midbrain here, and the rhombencephalon here, which is the hindbrain. So you can see that those basic divisions occur very early on. Here we have some rotating brains. And they're showing you the three parts of the hindbrain. And you notice that they are all to do with very primitive functions, but very, very important functions. Breathing, blood circulation, relaying movement, regulating breathing, taste and sleep. Cerebellum, important for motor coordination, precision and accurate timing. And in fact, the cortex, possibly, the big wrinkly bit, has been a little bit over-promoted in some senses. It is possible. It's a rather unpleasant experiment, and I don't think one would get ethical permission. You should not have ethical permission, I don't think, to do this anymore. But they did actually remove the cortex entirely from animals and noticed that actually they could still feed, they could still nurture their young, they could still walk quite happily, well, not that happily, but they could still do all those basic functions. All the things you need to live, basically, without a cortex. And some scientists make a joke now that the cortex is really just a duvet for the rest of the brain. Well, there's much more to it than that, but it's very important to understand that although the cortex is very important for our higher level reasoning skills, actually these other parts of the brain are very, very significant. That's what we're running on a lot of the time, and they feed in to a lot of what happens in our cortex. Now, the midbrain is probably the smallest part of those three regions. Important point, you do not need to learn these different parts in the smaller print for your test. Okay, probably want to make a note of that. Anybody who's not here won't know that, will they? I suppose they look at it on the video. But you don't need to know these little words here. They're words that are in smaller print. I think you should know the tegmentum, which is this part here, and then the tectum, which is just this region towards the front there. The tegmentum, there is a whole series of nuclei, and you know now that that means densely collected regions of neurons that are connected to motor control. And then we have the tectum, which is controlling and relaying sensory information. And so it's an important part of the brain. It's also important for reward function because it's dopamine originating from this region that can influence your reward response. It becomes very involved with that. We'll talk more about that later. However, the bits we're probably mostly going to be spending our time looking at on this unit are going to belong to the forebrain. There are regions that are not in the cortex, which are still part of the forebrain. They're subcortical regions. One of those is the thalamus, and that is a gateway to all of your sensory input. All of your sensory input, touch, vision, auditory, all of that information comes through the thalamus before it gets to the cortex. There's only one sense that doesn't go through the thalamus and goes straight into your cortex. Anybody know what that is? Smell, yes. Possibly that's why it's so evocative. Because one helpful thing about having a sensory gateway is that the cortex can actually mediate and influence that gateway. Whereas with smell, in you go. The hypothalamus is below the thalamus, and that is a good place for it because it's regulating our body environment. So if you're getting too hot, too wet, too cold, too dry, too thirsty, those sorts of things get picked up easily by the hypothalamus, information that's coming into the thalamus, and then the hypothalamus will take steps, if you like, to change your body response, help you keep cool or refreshed or whatever. So the forebrain is made of the diencephalon. We've just been talking about our diencephalon, and the telencephalon, which is otherwise known as the cortex. And below the cortex, we also have many subcortical structures that are included in the telencephalon, such as the striatum, the amygdala, the hippocampus. These are relevant to how we learn, very relevant. We've already talked about the thalamus here. We have the striatum, which is another subcortical structure around it, which is very involved with reward response, so our approach motivation. And one important part of that in the ventral area, so we call this the ventral striatum, and you know now that means below, yeah? So that makes sense because it's in the lower region of the striatum, is the nucleus accumbens. The more that activates, the more you want something. Sex, drugs and rock and roll, very visceral region of the brain. The amygdala, which is extremely important for emotional processing, is at the end of this sort of spiralling round structure here called the hippocampus. Hippocampus is very important for laying down your memory. Your memories may reside in different parts of the cortex, notably the temporal lobe here on the outside of the brain, but actually the consolidation, the laying down of those memories, the hippocampus is very involved with. So if you don't have a hippocampus, you might still have access to old memories, but you find it very difficult to collect new ones. So now we've just dealt with the forebrain. I think we've just dealt with all of that, actually. Cerebral hemispheres, I should make a mention of. So yes, the cortex comes in two halves, the left and the right. You can just about see through there in that diagram. It's separated by the longitudinal fissure, but it's connected by commissures, especially the biggest one, the corpus callosum. And the reason why I emphasize this is because I don't want people getting into the left brain, right brain thing. Okay? The left and the right hemispheres are communicating with each other all the time. Anything, even the most trivial thing you do, like picking up a cup of coffee, is going to involve it can involve actually most of your brain, but it will involve the left and the right hemisphere, and they'll be communicating with each other intensely, mostly through the corpus callosum. We saw a cutaway of the corpus callosum, didn't we? Did we? Did we not? No, okay, corpus callosum there. So that's a sagittal section. And you can see the corpus callosum there. We've had to cut that away, if you like, in order to separate the two halves of the brain. The human cerebral cortex is highly folded, double page of a tabloid newspaper in area. But the ridges that you see, we call gyri or gyri. The singular of that is a gyrus. So you may come across, I don't want to scare you too much, superior temporal gyrus. Superior means going up, temporal is one of the lobes, gyrus means a ridge. Okay, so that's the ridge. The valley between the ridges, so that's the gyrus on top, if you like. This is one of the wrinkles we've sort of taken out of the cortex. The gyrus is the ridge on top. The sulcus is the valley, plural sulci or sulci. Because it's Latin, there's no official pronunciation, but I don't really mind. You'll be writing it anyway. We have four lobes. It's another important thing to remember. We have four lobes. I've already mentioned frontal lobes. I've already mentioned the temporal lobe here. Remember that this is the front of the brain because we can see the cerebellum hanging under the back. We also have the parietal lobes here. Oh, a bit further back actually. Be there. And we have right at the back the occipital lobe. Very important for visual processing. So it's a bit weird that although the eyes are in the front, actually it's the back of your brain that does most of the visual processing. So that's how you can go blind if you have damage at the back of your brain. Now, you're already becoming very well armed here. So if I say to you, where is the superior temporal gyrus? You should know, okay? Because you can say, hang on, temporal, it's the temporal lobe. Superior means it's going up. Gyrus means it's a ridge. So actually it would be that there. I might test you on that in a minute. I've actually spoken about this. Just be aware of the fact there's hidden cortex. There is insular cortex, which I haven't shown, around about here, but also right between the two halves of the brain, the most important singular cortex is actually shown there. This part here, very important for paying attention, but all of it is quite involved with higher order thinking processes like decision making. Oh, it's colouring book time. So in front of you, you should have a piece of paper, and what I'd like you to do is to find the regions that are on the board and see if you can mark them on that piece of paper. I think what I'm going to do as well, actually, I don't know how many slides I've got left, but okay, no, we're just if you need a toilet break now, we're going to have a longer break in a minute, but if you need to rush out to the toilet, now's a good time to do it, because we are an hour into the lecture. So I'm going to give you 5 minutes to fill this in, and do feel free to go and use the loo or anything like that.
Which side? Is it this one or this one?
Well, I've been asked which side, but whichever side is the right one to choose to show the brain region. Okay. Oh, are there any more sheets? Any more spare sheets? Any more spare sheets? Oh, there's some spare sheets. Fantastic. To have a look and pass it up. Have a look at the brain, pass it around. You can have a hemisphere each. Have a hemisphere. Hemisphere and pass it around, have a look at it. Have a hemisphere and pass it around. Have a feel, as it were. Have a feel of a brain. I think it's a very good idea. Gives you a sense of its shape. Have you had a feel? That wasn't quite what I meant.
Didn't notice the brain behind it.
Do you want to have a pass it around and
So, inferior quadrant.
Yes. Yeah, no, well, I'm just in the middle of a lecture with 120 students, but they are doing something. That's why it's so noisy. Yes, is there any chance of making an appointment this week? It's yeah. I am. Yes, it has. Yes. That would be fantastic. Yeah, I'm not sure if we can be in. The slight problem is I'm not sure. So if there's any possibility of just leaving the money somewhere. Oh, okay. Oh, that would be helpful. Yeah, that would be great. Yeah, no, that would be brilliant. Okay, excellent. Thanks ever so much. Cheers. Bye. It's not always straightforward, I have to admit. Should we have a shot at them? Huh? Well, some of them are more useful than others, yeah. Ooh, inferior frontal sulcus. So let's have a little look at these. These are the answers, but the most important thing is you understand why. Inferior frontal sulcus. Frontal, okay? Frontal means a lobe. So that's the lobe bit. So first of all, you have to go to the frontal lobes. Inferior means down, doesn't it? It means towards the bottom. So now we're talking about those there. And then it says sulcus, so that has to be a valley. So we're looking at a valley towards the bottom of the frontal lobe, which is how you end up with probably something around about there. Yeah? I hope. Yeah. Something like that. Inferior frontal sulcus. So it's the lowest valley travelling along the frontal lobe. Did you get roughly there? Good, well done. The superior temporal gyrus, well again, temporal, that's the clue. It's got to be the temporal lobe. Yeah? Superior means towards the top, and it's a gyrus, so it's not a valley, it's actually the ridge there. So it's going to be along the ridge. So that would be a superior temporal gyrus there along the ridge. You okay with that? Did you get there? I've sent round some plastic brains because I thought it was quite handy for you to feel and touch these things in 3 dimensions. The dorsolateral prefrontal cortex is a bit of a dodgy one really, but you'll come across it so much. I'm afraid a lot of the time you'll come across the DLPFC. We do actually abbreviate it in that way, DLPFC, and you're just expected to know where that is. So let's take it one step at a time. Prefrontal means behind the front, just right behind the front. So in fact, it's around about, we're talking about this area, not right at the front, but around the front. And then it says dorsolateral. So dorso means up. So now it's probably around about there. And lateral means both sides. So it's that side and that side. So we're talking about an area, really. So dorsolateral prefrontal cortex is actually this region here. And it's a very important region for working memory. DLPFC. That's the information you're holding in your consciousness. Often has a lot to do with this brain region. DLPFC. Yes. So prefrontal means just, I didn't really tell you that, did I? But prefrontal means just behind the right at the front, basically. The superior frontal gyrus, well, and remember with those three, with the first two and with this one, I could have said left or right. But with the dorsolateral, because I had lateral, it was actually both sides. Superior frontal gyrus, the highest ridge, this is going to be along the frontal lobe, because frontal is the lobe, superior means on top, and it's a gyrus, so it should be a ridge. So now I'm looking for a ridge which is sort of quite near the top, up there. I'm not sure about these plastic brains actually, but anyway, probably around it. Oh, that'd be it there, superior frontal gyrus. I'll give you that one. Along the top there, superior frontal gyrus. Are we okay with that one? Did we get that one? Yeah?
So are these specific points, and just one of them, or there are several that can be referred to?
They should be specific points. Yeah, but, you know, everyone's brain is a little bit different, but most people will have a superior frontal gyrus. Yeah. But some will be longer than others, some will be a bit more wrinkly than others. And then we have the anterior cingulate. And the only way you're going to see the anterior cingulate is to do a quick hemispherectomy and divide your hemispheres and look inside. And that's going to be anterior cingulate. Anterior means near the front. So it's going to be the island that's between the hemispheres, but anterior means the front of it here. Yes?
If the hemisphere isn't specified, do you assume that it's bilateral?
Well, it will. In a technical report, they will either say bilateral, or they will say left or right. They'd be picked up by the reviewers if they just said it's in the anterior cingulate. Because they say, well, which anterior cingulate? So it's a good point, because there's always going to be two of them. In fact, with all of those except the dorsolateral prefrontal cortex, but even with that you could say left dorsolateral prefrontal cortex or right DLPFC. So with all of these there could be two of them, left or right. And that's also true for a lot of subcortical structures like the amygdala. You can have left amygdala or right amygdala. Right, shh, shh, shh. Ladies and gentlemen, please. Hush. Thank you. I did used to be a teacher. So I come fully armed with classroom management strategies. Brodmann areas, I need to talk to you about these because you will come across them, but there will not be any tests. They will not be tested, okay? But you need to be aware they exist. Because they won't just say dorsolateral prefrontal cortex. They will probably say left dorsolateral prefrontal cortex brackets BA8. And you think, BA8? What is that? But you need to know that's Brodmann areas 8. And then you can go back to this slide, or you can go to Wikipedia, which is where I got the diagram from, and you can find out that Brodmann area 8 is just up there. Okay? Why do we have Brodmann areas? Well, Brodmann areas arose from looking at the microarchitecture of the brain, noticing that the granularity of the brain changes over particular boundaries. And therefore, Mr. Brodmann, or probably Professor Brodmann, decided to divide up the brain into all these different areas. And we do find, interestingly, that function is broadly impacted upon by those boundaries. So it makes sense sometimes, makes more sense sometimes to talk about BA8 than it does dorsolateral prefrontal cortex. People haven't decided. The truth is the cortex is plastic, and so the way in which the cortex organises itself is very different from one individual to the next. Not very different, but it's different. So functions don't keep very happily to always just like one clearly defined area in the brain. Not when you look across individuals. Yes, well, I suppose I should say some very famous Brodmann areas are 1, 2, and 3, because that's primary somatosensory cortex. So information coming in from your touch and your feeling is going to be there. Bang next to that is the motor cortex, BA4. And of course that's very helpful because you need to have your incoming senses being processed right next door to your motor commands going out, because it's more efficient for that processing to take place. That's what's organised like that. And then we have, supplementary motor area and premotor cortex also involved with processing. Oh look, dorsolateral prefrontal cortex, Brodmann area 9 and 46, but 8 can be included. 9 and 46. Anterior cingulate cortex, Brodmann area 24, 32. So just be aware of what these Brodmann areas are. You've always got this slide to go back to find out what they're talking about. Let's talk about, and I've already started talking about it, but let's start talking about where function is localized in the brain. And we have to be very careful here because all things like memory, attention, emotion, they are much more complex than simple sensory or motor function. We can see relatively clearly where motor function and sensory functions are in the brain more clearly than we can some of these broader, more complex forms of cognition like memory, attention, and emotion. They tend to be distributed in many areas of the brain. And no one part of the brain is responsible for these. And we have to be careful not to go back to phrenology, where we were looking for things which make sense to us as a human, because we tend to mentally put them in a particular category. And this is what psychologists have been doing for decades. Intuitively we've felt that this belongs in a certain box, a different category to this one, because that's how we talk about it in everyday life. But then we come to the brain and we find out that actually we don't have a secretiveness region of the brain. We don't have a hope region of the brain, or a wonder region of the brain, or a wit. What else we got? Parental love. So, or an acquisitiveness region of the brain. So the brain is organised in much more primary ways than that, much more fundamental ways than that. And whenever it comes to something which, to us, feels like a very important human category, you're usually going to find it very distributed across the brain. So please don't fall into the phrenology trap. For example, I want you to think about reaching out and touching this cup of warm, delicious soup. Okay? Just think about that for a minute. When you started thinking about it, you already start activating your motor cortex and your somatosensory cortex. Those are topographically organised, which means that your fingers, your hand, your arm, etc., as they are organised on your body close to each other, so regions involved with the processing of that in your brain are also organised together. It's good to have the input, the feel of those regions, and the motor control of those regions close together. And you can see that that's one of the first things that activates. So already we have activated the somatosensory strip and the motor cortex here is beginning to activate. We also are beginning to have parietal cortex activated. The information to make that happen in terms of the touch, remember that's got to have gone through the sensory gateway of the thalamus as well. Okay, so I can't show that on this brain because it's underneath the cortex, but that will also have been activating. So information from the thalamus reaches the somatosensory cortex, from there the motor cortex, and also there into the unimodal somatosensory association area, allowing the shape to be formed from touch. Separate information from each of those fingers will have reached the brain. That has to be joined together to give you a rough idea that you're touching the shape of a cup. So that association will happen in a unimodal association area, which will be in the parietal cortex. From here it feeds into premotor regions. Premotor is for planning. Because actually, although you've been thinking about reaching out for it and such like, actually you haven't started doing that. You're just beginning to think about planning to reach out for it. So premotor cortex will begin to activate. There it feeds a preparing movement in respect to feeling and the shape to the higher order, more posterior unimodal associations here. So already we are beginning to get extra information coming into the premotor regions from the parietal cortex, which is saying there is a cup and you're going to have to bend your fingers properly in order to be able to lift this cup up. And now we have converging with other information from other sensory systems into multimodal areas, which are also more in the parietal cortex, but more posterior, where you're combining that information about the shape with the fact that it's actually quite warm. And maybe you could even smell it. So all that information is now getting combined into the idea that it is a cup of soup. And that has to bring together all those different bits of information from different modes, and that's likely to happen in the posterior parietal cortex here. And of course, at the same time, you're remembering the last time you had this soup, it was very nice. And so that's activating memory here, which has been stored in your temporal cortex. And all the time, you're trying to concentrate, and that's probably going to be activating your cingulate cortex. And your parahippocampal cortex as well is very important for bringing back those memories into your cortex about what happened last time. Where do you stop? I could go on. I haven't even mentioned the fact that the visual cortex is going to be activating because you're actually seeing the thing. Okay, we haven't talked about that. So there's going to be all that information coming from there. And then at the same time, you're trying to make a decision, actually, how many calories has this soup got? Which is probably going to be the frontal cortex. So all of that is happening at once, and if you could see brain activity, just in that 2 seconds, you would see this shimmering of activity all over the brain. All sorts of different functions, complex functions and reasonings, and conceptualizations taking place. How do we see function, therefore? Well, for one thing, we tend to ask people to repeat the same thing again and again and again, because, essentially, we can't have people picking up cups of soup. It's just too complicated. So we ask them to do something much, much simpler, like look at this dot, or something like that, something very simple, or move your left finger, Please don't do anything other than moving your left finger. And you do that like 100 times. And of course they are doing lots of other things, but hopefully they all sort of cancel out after a while, and you just get left with the brain activation for moving your left finger. And how do you pick up that brain activity? Well, one way of doing it is to ask them to ingest something that is radioactive, which is going to attach itself to parts of the brain that require, for example, glucose. So you could ask it to, you could put in a radioactive messenger attached to glucose, and then when the neurons are activating, they'll need energy and they'll take in that glucose, and you'll see a collection of that radioactivity in that particular region of the brain. And then you just need to scan the whole brain to pick up that radioactivity, and that's called positron emission tomography. You can't do it too many times because it's not terribly healthy to be ingesting radioactivity. It's difficult, therefore, for people like me to use it, because I don't want to use up the 5 times that you should do it in case you need to have a PET scan done in hospital. However, the spatial resolution is not that good. You can see it's quite a fuzzy image you get of the brain. The temporal resolution is terrible, so trying to work out when something has happened is not very good either. Apart from that, it's great. No, what's really good about it is the fact that you can attach your radioactive messenger to lots of different molecules that can be involved in different ways in different brain processes. So you could have something that was particularly dedicated to cleaning up dopamine. And if you attach your messenger to that, then wherever you get a lot of dopamine, you're going to get a lot of chemical radioactivity. So it's quite useful for those specific functions, but it's not good for finding out exactly where, and it's not good for finding out exactly when, and there is a safety resolution, a safety issue. Electroencephalography is EEG, otherwise known as, And here you're recording the activity outside of the brain using a hairnet made of electrodes. And that electricity, that change in the electric field, is actually being brought about directly from the neural activity. So the really nice thing about that is that it's very responsive. So you have very good temporal resolution. So obviously as soon as your neural activity is increasing here, you're going to get a change in electrical activity, and you can pick that up almost instantaneously with EEG. So it has fantastic temporal resolution. Spatial resolution is not so good. It does improve with the number of electrodes, but there is a limit. And also, obviously you can't pick up electrical activity subcortically. It tends to be more of what's happening on top of the cortex, if you like. You can use it with specific behavioural markers. So we know, for example, that if you get two stimuli and there's a difference between them, then you get a particular type of response from your EEG. So you can use that to find out whether babies even can hear the difference between two audio signals using an EEG cap on their head. So that's quite useful, having those markers. When we do that, we call them event-related potentials, and we say we're doing an ERP experiment. We also have oscillations in the brain. We notice that this electrical activity can be analysed in terms of certain frequencies. You know, when it all comes out, it's all a mess. But if you filter them, you can see that there are certain very strong frequencies. One of them is alpha, which appears to increase with relaxation. One of them is beta, which becomes stronger when you're anxious. And one of them is theta, which means that if you're getting a lot of theta, you're probably very relaxed, you're meditative. It's been associated with creative states. So when you've got a lot of theta, but in fact you're awake enough to be doing something, then that's a good sign of creativity. Usually it tends to bode well for creativity. So people often measure the theta-alpha ratio or the alpha-theta ratio to find out if you're in the right place to be creative. And by training yourself, because you can actually buy kits to train yourself to produce more alpha-theta, less alpha-theta, isn't it? Less alpha-theta. Then, so you've got a lot of theta, but not too much alpha. Then you can actually increase your creativity. So musicians at the Royal College of Music trained themselves using this, and in their performance examination they were able to increase their final degree result by a whole grade. So those who would have got a 2-2 got a 2-1, and those who were on for a 2-1 got a first. So it's very interesting, and they've started doing trials with this with children in schools. This is my favoured weapon of choice, functional magnetic resonance imaging. So blood flow links to neural activity. Active neurons consume oxygen, but the increased blood flow occurs without matched oxygen extraction, causing an overall increase in blood oxygen. And you can pick up that increase in oxygenation by the change in the properties of the magnetic field. So you put people in a magnetic field, and to find out what the properties of that magnetic field are, you pulse it with a radio frequency. And the way that it relaxes back depends on what is in the magnetic field. And they've found that one of those properties of whatever's in the magnetic field that makes a lot of difference is the amount of oxygenation in the blood. So that gives us a signal whereby we can say where is functionally active in the brain. So we put people in a very high magnetic field, and we will be looking for volunteers this year. It's about 20,000 times the Earth's magnetic field. And then we pulse it with a radio frequency. And we have to ask you to wear ear protection because it's a bit noisy. And it's a little bit of a confined space as well. Apart from that, it's lovely. But you do get a lovely picture of your brain out of it. So it has very good spatial resolution. Temporal resolution, not quite so good. But there's no health limit either. You can have as many of these as you like. I mean, I've had about a dozen of them. Well, I'm perfectly normal, aren't I? So this is our particular facility up on the hill, the Clinical Research and Imaging Centre called CRIC. It was opened a few years ago. And you'll be able to, you actually can look that up on your website and find out more about what they do there. So, we're about to have our break. You have learnt about neurons and other cells. You've learnt about neural networks, how communication occurs within them, the action potential, that's that bit of electricity that goes down the axon. You've learnt key orientation terms, they are very important. You've learnt there's 3 parts of the brain, the 4 lobes of the brain, and you've learned about some subcortical structures. That means structures that are underneath the cortex. You know about Brodmann areas, even though you will not be tested on them. And you know about some associated functions of different brain regions. And a little bit about how we find out where different parts of the brain become active using these different techniques of fMRI, positron emission tomography, and EEG. Why did I pass around? What I try to do during these lectures a little bit is I just try to tempt you into thinking about what the relationship is between what we know about the brain and teaching and learning and education. I asked you to touch and feel a brain because I wanted you to remember its shape. Vision and touch are actually processed together in very similar regions of the brain. So we can see that, in fact, there are some regions that completely overlap. These green regions here completely overlap in terms of how you process touch and you process vision. So we know that by seeing, but also by touching, that really reinforces some concepts, particularly, for example, shape. So because I wanted you to start thinking about the shape of the brain, I wanted you to touch that brain. If you haven't touched it yet, please touch it now. Someone's got a brain near you. And that should help you to remember it more. So it's an example of multimodal processing. We didn't know that before neuroscience. So audio-visual processing, where you're processing sound and vision together, is actually very different to that. It has to be brought together in the brain conceptually, and you have to do some thinking in order to do that, whereas this happens automatically. So not all multimodal processing is the same, and you need to think about how different modes combine in the brain in order to use them effectively in the classroom. Why did we play Simon Says? Because action and cognition, those brain processes, actually evolve together in the brain. This is called embodied cognition, and it's a whole region of thought that makes us focus more on action as a way of understanding our mental processes. And by combining action with mental processes, we can learn more effectively, and that's called the enactment effect. So when you do something, you're more likely to remember it. So that's why I asked you to play the Simon Says game. And imagining a cup of soup is important, because when you imagine a cup of soup, you activate almost as much of your visual cortex as you would if you actually picked up the cup of soup yourself. Which really points to what an important tool, potentially, visualization is in education. And probably we don't use it enough. But visualization, very powerful way of conceptualizing, activating almost as much of the visual cortex as if you were actually seeing it. Okay, just a few things to think about there. We are now going to take a break. That break is going to be 15 minutes, and we're going to come back here at 12:45 for a 1-hour lecture on development. Okay? Are we all right? Any questions? Before we go, hang on, just before we go, because I don't want to leave this topic behind quite yet. I just want to make sure we've dealt with any questions. Yes?
What happens above 30 hertz? Does the brain not resonate?
Oh, there are, I think it's delta. There are other waveforms that I haven't put on there. I'm not an expert in EEG, but that might be delta waves, I don't know. But I think there are other waveforms. Yeah.
Anxiety.
No, I think anxiety is measured more by those ones, I think, as being opposite to relaxation. So you'd expect a change in alpha for anxiety, I would have thought. Yes? Hang on, people. I know this is very difficult. I know you're dying to get out for some fresh air. Me too. But just hang on there.
When the wavelength hits 30 millivolts,
Yeah.
the potassium channels open.
Potassium channels open, yes.
Already open, and it opens further.
Yes, yeah, opens further, I should say, yeah. It's flowing at so the positive ions can come in. Yeah. Sorry, at the back, gentleman there.
Wasn't there a recent issue with fMRI and software?
There's an awful lot of issues with my software, but that's me using it, I think. But I don't know. I think one of the problems was that radiologists use a different left-right notation to researchers with fMRI. So it's quite easy to get left and right mixed up, and that's quite important if you're using it for an operation. And I know that 10 years ago when I was on a course, the surgeons were actually doing a test to find out which was left and right hemisphere, if you like, and they would actually sort of put in a little screw or something to actually indicate which side of the brain needed to be operated on. So I think things have come a long way since then, and that the software now, I think, follows the right conventions. It's one of those historical things that the radiologists and the researchers were using opposite standards, essentially, for left and right, which is a bit unfortunate. I think that's probably what it was. Yes?
So I just want to double check what lateral means exactly. What would be the lateral? Is it away from the...
Yeah, lateral means same side, but so it's the same side. So essentially, if I said I moved my right hand and I had lateral activation, it means that it'd be on the right hemisphere. Bilateral means both sides. Yes. Yeah. It means that basically you can't. So spatial resolution means it's the smallest distance that you can pin something down to. So you know, like when you're taking a photograph, if you don't have many megapixels, it becomes very blurred, and then you get poor spatial resolution essentially because you can't see which is where. And that makes it very difficult to know where in the brain regions that particular activation is. Yeah. Yes.
When you said rigid part, that's when you rigid activity. Where's the rigid? Is that the part?
The ridge is between the wrinkles that are marked on there. So it's the wrinkles that are marked usually on a diagram like that, or the sulci, and the gyri are actually the bits between the wrinkles.
Gyri is between the wrinkles and
between the lines. And the sulci are actually the lines. So it's the lines that are actually usually marked on those where you see the wrinkles. Anyone else? Okay, I think I've gone and taken up some time now. Can I just ask, though, please, brains, could you be very nice and bring down my brains? If you can find another part to it and make it into a whole brain, that would be fantastic. Otherwise, just bring back the half a brain, please. Thank you.
Do you need us to sign it?
It's 40. 10-2. I think that's the olfactory bulb, I think. Yeah. So in mice and rats and things, it's much bigger.
Sorry, just a question. Are we going to get the slide of development?
No.
By email or?
What are you going to get? The slides?
The slides.
Slides are on Blackboard.
Ah, okay.
Definitely. Yeah, there's a register. Who's got the register? Anyone seen the register? Oh, is that it there? No, that's my spare one. Yeah, be careful of this wire here. Thank you very much. Who's got the register? Anyone seen the register? Oh, we'll have to do it afterwards.
Yeah, so I think what to find the part here.
Oh, I see. You're the gentleman. Is that Luke? Has it? Well, it should be. It looks exactly like that. It should be around there somewhere. Has anyone seen the register? Can you make sure no one falls over that?
Sorry, I know you want to get off.
I'm looking for the register, actually.
Register webcast.
Who's got the register? Register? Anyone seen a register?
The register was behind somewhere. I passed it.
Funny that thing's... Oh, there it is, right. It's that one. And I'll have a brain as well. I'll have the brain. Anyone not signed the register? There's loads of extra people on there. I thought it was crowded. Hello.
Hi there. So I'm going to tell you. You tell me if it's right. So this is the gyrus.
Let me just mute this. So this is a gyrus here, right? And that's a sulcus.
Okay. When you meant the rigid part.
This is the rigid part or the
region. The
ridge.
Yeah, ridge. I know this is the ridge and this is
sulcus.
Sulcus.
Yeah.
But the cortex is folding and they are the same thing. So cortex and
Yes, it is both cortex, but they use that to identify.
two different regions.
Yeah, they identify in that way. But you're right. There's no region. There's no reason I know why a gyrus should behave differently to a sulcus. But because we're trying to identify a particular part of the cortex, they use these ridges and sulci just as a way of saying where it is.
Is rigid and sulcus the same thing?
No, that's a ridge. That's a sulcus.
The material is the same.
Gyrus and ridge the same thing.
Yeah, that's a gyrus. Yeah, gyrus and ridge is the same thing.
Okay, that's what I want to know.
Gyrus is a ridge.
Okay. Sulcus is a valley.
Gyrus.
And a sulcus is a valley.
That's all I wanted to know.
Yeah.
Thank you.
Okay. Well done.
The
Sure.
myelin sheath.
Yeah.
I understood that it also had a function of speeding.
Sorry, the which one?
The myelin.
Oh, the myelin sheath. Yeah.
Yeah, I always read just in earlier studies.
Yeah. No, it does. It does. It does. Yeah. It does speed up.
Do we need to be able to know that, or? I mean, because you talked about it.
Not
being
really.
an
I was only talking
insulator.
about it. Well, I think that's why it speeds it up. I think because it's an insulator, it does speed it up.
One node of RAN VA to the other. Is that how it works?
Yeah, because it can't get out anywhere else, so it sort of shoots down.
Okay, so the insulation is to speed up.
Yeah.
Right, and if it breaks
Well,
down,
and also to
then
keep the keep it in as well.
Oh, okay, so it's got double function.
Yeah.
Okay.
When you're talking about the myelin sheath, is that made of fats? Is that correct?
I think so. Yeah.
Yeah, and then depending on the quality of fats in the brain,
that's
when you can get the charge escaping. That's what you're talking about, isn't it?
Yes, if the myelin breaks down, then you will basically lose the efficiency with which the electric potential goes down the axle.
Yeah, that results in
so it'll be slower, but there'll be loss as well. Yeah.
Excellent.
Okay,
Thank you very much.
Okay, nice to meet you, Lou. Are you on the neuroscience course then?
No, I'm doing the MSc teaching learning, so it's a bit of a kind of a hybrid.
Ah, right, interesting.
But,
maybe you should think about swapping to neuroscience then.
I've only got this much left in the dissertation.
Oh,
so
have you?
right,
never mind.
almost done.
I'll let you off.
Yeah, I'm waiting next week, but I'll see you.
Okay.
Thank you.
Hi, I just wanted to ask where I could find the idea of the Royal College of Music, where they increase the creativity.
Yeah, when you go on the Web of Knowledge, Williamon is the guy, but also Griselier. I can write it down for you if you want to. So one of them is a scientist and one of them is a musician. Williamon's the scientist, is the musician, and Jay Griselier is the scientist.
All right, cheers. Thank you.
Okay. Hello.
Your break as well.
That's all right. So get these brains away.
About the practicalities of basically I'm pregnant with twins.
Oh, congratulations.
Thank you.
Double trouble.
Terrifying as it is. And the date of my induction is actually beginning of November. I was wondering. Probably not.
Oh, sorry. Your induction. I thought you meant academic
Induction.
induction. You mean your birth induction?
Yeah.
Okay. You can't take it early because I think there might be. Yeah, I think there might be difficulties with that. What you can do is take it late.
Okay.
So when you're feeling up to it.
How late? Because obviously. I mean, I was hoping to submit the poster earlier as well as the assignment. That's my plan. But,
well, would you possibly be able to do it after Christmas?
probably will have settled into.
Would that be good?
Yeah.
Yeah, because I'm actually in Geneva anyway, so it'd be easier for me to organise it after Christmas. If you just contact me again and say, "Do you remember me? I was having twins."
I'm really keen to this is one of my.
I've got the birthday cake, so here is a piece.
Oh, this is very kind of you.
Yeah.
Thank you very much. Okay.
Okay. Yeah.
So, are you a teacher or a psychologist or?
Working for 16 years. I'm social teacher,
Right.
academic mentor.
But you want to become a psychologist?
Something. I don't know what I want to become yet. I like the aspect of recognition. Anyway, that's all I wanted to ask you. So.
Okay. But that's fine. I can arrange that after
I might
Christmas.
be able to do it on the 7th, but it's a bit touch and go.
Yeah, no, let's do it after Christmas.
Depends.
You know, it's not very difficult.
No.
So it's not very difficult.
And I can submit the other
Yeah.
poster and assignments earlier. And the suggested dates.
Yes, you certainly can. There's no problem with that. It's just a test. If you can do that that would be great. Could you email me? Just let me know.
I will.
Email me to remind me what we've decided. Just email me and say thank you for letting me do the multiple choice test after Christmas, after my twins have been born, and then I'll have it in my memory.
Have it in your memory.
Yeah, do you know what I mean? I'll have a look back on it there, and then
Activate your hypothalamus.
that's right. Activate my hippocampus. Don't trip over that, really. That'll be a disaster for you.
Yeah, I'll send you a short email.
Yeah, that would be great. And what's your name?
Alina.
Alina. Pleased to meet you, Alina. Thank you very much.
Thank you, Paul.
Half a brain missing somewhere? Oh, one half a brain. I'm missing half a brain. Yes, it's over there. I got it. Design and technology. Yeah. At Rolf Allen School in Bath. Yeah. Hey? Did you? Well, it was quite a while ago. Frank Thomas was the head teacher at the time. What's your teaching then? What do you teach? MFL. Jackie Haggett. Well, no, she was maths. Can't remember now. It's quite a while ago. When were you there then? When were you there? It'd be quite recent. Yeah, it's probably about 20 years ago for me. Oh, thank you very much. Trish. Yes, Trish Vowles. She was there when I was there. Yeah.
I'm just wondering, you know the next lecture we're going to have now?
Yeah.
Are there slides online for it?
There are.
Are they up already? Are they in
They are.
section 2 folder?
They are.
So the title of it is Learning.
But they're in section 4. No, sorry, in section 4, not section. For some reason they're down because I moved it. Yeah, the title is Development.
I've got some, but at least the slides are
Right. Right, there are no more brains, are there? No, I don't. Anyone feeling strong? Anyone feeling strong? Looks like a strong chap. So you could take one of these, and I could. Yeah, and there should be some in the toilet. I'm sorry I haven't got any. Right. Might have worked. Thanks. Disrupting is so vague. It is an effort to localise the function. There's quite a few more as well. There's magnetoencephalography, which I haven't mentioned, and infrared as well. There's so many. I just thought I'd pick three. Yeah. Whose birthday is it? Quite nice. One more brain. Okay, let's get back to it. I want to do this in an hour or less. Hopefully let's do it within the hour. It is of course prohibited to bring any food or drink into the lecture theatre. So if I had done such a thing, I would make sure that I had taken out any evidence of such. Having just YouTubed myself eating birthday cake, I realise. Happy birthday, Maria. Lovely cake. Right, so development. I think the most important thing about this, I'm going to be talking a little bit, I'm going to go through in a very hand-waving way, very rapidly, through how the brain develops. First of all, none of this is going to be assessed. So why am I doing it? It's really because I want you to get a general impression of brain development, because there are some important messages about how we should think about the brain in a broader sense. And it's those messages that I really want you to hang on to, rather than knowledge about the embryonic disc or anything like that. If you're going to buy a book on development, I think Developmental Cognitive Neuroscience by Mark Johnson is really good. Highly recommended, particularly, in all senses really. This is definitely the book to get if you're interested and you want to know more about this. Now this is important because actually I almost put this slide up first today on the basis that maybe you were only ever going to remember the first slide. So I thought maybe this should be the first slide, really, to get you thinking about the way in which scientists consider behaviour. There is an assumption, and this is the scientific model. And I don't know if you're going to agree with it or not. I don't completely agree with it myself. But this is how we must think about things scientifically. This is the best we have at the moment. Development is an interaction of genes and the environment. So we no longer talk about, is it nature, is it nurture? You know, the way in which we have sex differences between boys and girls, for example. Should we think about those in terms of nature, or should we think about them in terms of nurture? Well, in terms of sex differences in cognitive function, we can think about that as an interaction of their genes and the environment. It's an interaction of nature and nurture. It is impossible, really, to have any phenotype, that's the outcome. It's impossible to have any outcome in terms of a human being or any other sort of animal, or even a single cell, purely in terms of genes. If you just had the genes, nothing else would happen. Similarly, it would be impossible without, if you didn't have the environment. So you have to have both of these things interacting, and that's how we think about them. So forget about the nature or nurture argument. This is all about nature interacting with nurture, genes interacting with the environment. And the basic assumption is that the organism that you get, so you here now, for example, are completely a product of your genes that your mum and dad gave you and your history. That's what you are, okay? Nothing else. Now, of course, if you have religious interests, you might disagree with that. And I'm not really here to argue that. All I'm here is to say this is the way that the science thinks about it, okay? It gets worse in a way, because if you think that's a bit confining, because now we say actually your behaviour, what you're going to do next, is simply a product of your phenotype, the person that is here now, and the present context. Which means that if we know enough about the present context, and we know enough about your genes, and we know enough about your history, we can predict everything that's going to happen for the rest of your life. And be completely determined, all right? That's the scientific model. We don't talk about anything beyond that. I mentioned religious issues, because you might want to put God in there, or gods. But actually there's something else in there which I always feel very uneasy about leaving out. Anybody want to hazard a guess as to what that might be? Anybody think? Sorry? Ethics, responsibility. You're getting closer there, responsibility. Free will. Where is free will in the biological science or the cognitive neuroscience of who we are? It's actually a very problematic area. An awful lot of neuroscientists do not believe it exists. Okay, they say, no, this is it. Everything that you think is a freely willed decision, actually that's just post hoc justification. It's justification that's happened after the event. And there are experiments to show this. So, for example, Antonio Damazio's experiment that you'll be coming across in the emotion lecture, it shows that people's body starts responding before they're consciously aware that they're going to change their mind about their strategy in a card game. If you ask them what they're going to say, no, I'm not going to change my strategy, but already the body is actually reacting. And then, of course, afterwards, yes, I decided to do that because blah, blah, blah. But no, actually, it's your biology that made the decision. Your consciousness is just, well, what is your consciousness? This is the trouble, because free will, if we're going to talk about free will, we have to talk about consciousness. We have to talk about you. Where are you in your brain, right? And this is where I start going, oh, this is an introductory course to neuroscience. I cannot cope. However, Richard Parker, he's going to try to tackle that a little bit more. He's going to be talking to you about some of the issues involved with thinking about consciousness in that lecture. I think it's about 3 weeks' time. Okay, so that's the dogma, if you like. In theory, every action taken could be explained in that way. Actions are just a product of your present context interacting with your genes and your environmental history. How does it all start? Well, it starts off with a little fertilized cell, and pretty soon the cells multiply according to their DNA, and they start differentiating, which means they start going into different types. And at day 5, you can see that we're starting to get a sort of a cavitation. There's sort of a working space has sort of emerged in the middle of this. We've got a sort of protective outer layer and something. There's a space in the middle for something to happen. And what happens is that you get further differentiation into 2, when you've now got 3 cell types, and you've got these 2 spheres which are impacting on each other, and between them we have this 2-layered disc, a bilaminar disc. And then further we find that the outer cells hatch and implant into the uterine wall, but it's really this bilaminar disc where an awful lot happens. So that's the thing we're going to focus in on now. If you're a bilateral animal, you have symmetry, okay, and not all animals do. There are crabs, for example, that have a very big clawy thing here and a smaller one there. But if you're symmetrical, you have a primitive streak here, which appears, little line, and then further differentiation of new cells here forming between those 2 layers. So you end up with actually a 3-layered disc. And between the top and the bottom layer, we have the mesoderm layer. And these cells create something called the notochord. And the notochord is really important because ultimately that's what's responsible for your central nervous system. On the ectoderm layer, which is the top bit here, opposite the primitive streak, we have the neural plate forming. The neural plate. So this is a diagrammatic form of what you've just seen. So you sort of say, well, how has it become a disc? But remember, you've got 2 spheres sort of impacting on each other, and that makes a disc, doesn't it? Now this notochord releases the proteins that drive neurulation, the production of neurons. And this ectoderm will become skin, the endoderm will become your internal organs, and this mesoderm will become the muscle, the bone, and the connective tissue. This notochord is going to be absolutely essential for producing the neurons in your central nervous system. And what happens now is we get this thing called invagination, or folding inwards. So here we have the 2 spheres, if you like, and we've got this sort of interesting layer here that we're very concerned with. But now it folds inwards. It folds inwards like that. And it starts forming this little sort of bulge. And if we just take a cross-section of that, we can see now we've got this notochord forming here, and the bulge is coming in like that. And this bulge here once it's sealed at the top, it becomes the neural tube, and that's what forms your brain and your spinal cord. Whereas this crest at the top actually forms the peripheral nervous system. Now that neural tube starts outputting neurons, and neuroblasts and glioblasts, and those are cells that produce the neurons and the glial cells. One end of that neural tube becomes a series of repeated units, bit, 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 and starts looking very much more and more like a spinal cord. The other end develops some bulges, and those bulges become your forebrain, your midbrain, and your hindbrain. Ultimately, it becomes the finer structures as well. But quite early on, within a few days, you can start seeing this basic plan of the brain, this tripartite plan in 3 parts, appearing. And it's very similar for a chick as it is for a human. It's quite difficult to sort of see the difference at that stage. That's going to become your forebrain. That's going to become some of the subcortical structures under your forebrain. This will become your little midbrain. This will become your hindbrain, including your cerebellum. So the ectotherm thickens to become the neural plate, which then folds to become the neural tube. The neural tube becomes the cerebral cortex down here. That's the forebrain. It also starts forming the midbrain and produces proteins for producing the hindbrain as well. Now these cells that are coming out of the neural tube have to move. They have to move and they have to find their way to the right place in your brain. We've got neurons and we've got glial cells. How are they going to move? Well, the 3 things essentially they do. They proliferate, in other words, they're born. They migrate, they travel, but they're also at the same time they're differentiating into different types. And depending on the type of cell, it has to find the right place for itself. So then you sort of think, well, that's very intelligent. How does it know where to find the right place for itself? They either migrate past the older ones. There's 2 ways in which they can travel. They can either overtake the ones that are in the way, if you like, overtake them, or they can actually displace them and push them further away. And in so doing, using those 2 different methods of moving, cells of particular types start developing in particular regions of the brain. And that's how, for example, in the cortex, you start getting up this multi-6-layered structure of the core humans, made up of different neurons. So this is actually a section through a human cortex here, and they're actually different regions of the cortex, because they'll have a different layered structure depending on which part of the cortex you're looking at. And can you see the layers? Is that clear to you? Can you see that there's a sort of you could draw a line there, you could maybe draw a line there, draw a line there. And there's about 6 layers you should be able to see in there. So what are the key changes that happen? Well, one of the most important changes, or some of the most important changes, happen prior to birth, which is the arrival of almost your entire neuron set. So just in the first 3 months of conception, mum is basically responsible for producing the entire neurons, the entire set of neurons, and we're talking about 86 billion neurons that child will have for the rest of their lives. We used to think it was the entire set. Slightly good news on that front, but pretty well it's most of them. Slightly good news is that we do carry on producing neurons throughout our lives, but we've only just discovered that and we're not quite sure what it means. But most of the 86 billion are created in the first 3 months after conception. That's 250,000 a minute. So I don't know if it's got anything to do with morning sickness or not, but it sure seems like a good reason why it can make you ill, because that is an incredible feat by anyone's standards. Or feel ill, should I say. So The other things that happen, of course, is the increases in weight, increases in synaptic density. Now, what do we mean by synaptic density? I've put that phrase on you, and I haven't told you what a synapse is. We talked about connections, and the connections being really important between one neuron and another, because that actually is how we learn, when those connections change their efficiency. Well, another word for a connection is a synapse. And synaptic density means the number of synapses in a particular volume. So if you've got a lot of connections, you've got a high synaptic density. And synaptic density will increase greatly, and it peaks in different regions of the brain at different times. So 10 months after you're born, there is a massive peak in your visual cortex synapses. But actually other parts of the brain, like the frontal cortices and the parietal cortices, actually those parts will carry on for many more years producing neurons, and as well as producing neurons with these waves of proliferation, these waves of production of sorry, not neurons, synapses. Production of synapses, as well as waves of proliferation of these synapses, there are also waves of pruning back of synapses, which we also think are very important, where you lose a lot of synapses. And although those processes happen quite early on in the first few years, in the visual cortex, in the parietal cortices, the frontal cortex, actually that carries on till you're around about 19. So it's not over by 10 months by a long shot. And of course we also see this increase in insulation, the increase in myelination around the axons. And yes, this is the point I was making here. Some changes carry on into your teens, and also the myelation processes continue into teens as well. PFC, so you have to be very careful with me, because I put in little things like that. You'll find this is very annoying in reports. So PFC, hang on, he hasn't said what PFC is. Anybody want to make a guess? Prefrontal cortex, very good, yes. Prefrontal means just behind the front. Prefrontal cortex. How do these different neurons know where to go, though, when these neurons are being created? We're going back to talking about neurons being created now, having talked about synapses. What sort of interaction is responsible for cortical laminate structure? That's the layers that we have in the cortex of the brain. Well, one process that we know of are the glial cells. It's possible that the glial cells actually play quite a role in guiding the path of the neuron, acting rather like training stalks in a garden. If you're trying to train your peas to grow in a particular direction, then you put in sticks in the ground. And these glial cells can act a little bit like that. So to some extent, this travelling of the cells to their appropriate place in the brain could be considered innate. Now, innate, when you use innate in your essay, right, my alarm bells will immediately go off, right? Innate's a very dangerous word. There are some very dangerous words in this unit. Innate is one of them. Cause is another one, okay? So you always have to be very careful when you use those words. What do we mean by innate? Well, that's kind of what I'm talking about in the next few slides, how careful we have to be about this. Is the fact that we end up with Brodmann areas, with different cells in different regions, that we end up with this map of the brain, is it innate, or is it to do with experience? You know, is it nature, is it nurture, if you like? Well, Colin Blakemore and Molnar, Colin Blakemore's Sarah Blakemore's dad. Sarah Blakemore's the teenage neuroscientist, the neuroscientist who deals in the teenage brain, I should say. But her dad, Colin, placed some visual thalamus on a petri dish. Now this is thalamus, which obviously, you know, it's being kept alive, but it's not part of an organism anymore. And they put it on a petri dish with some visual cortex, and they noticed that the thalamus afferents are immediately invaded, and they stopped exactly where they were supposed to stop, at layer 4. So amazingly, you know, these thalamus, and remember these are afferents, so they're actually, you know, they're trying to wire up the thalamus and the visual cortex. They knew where to go. obviously there's no environmental input at all into that, because it's not in a functioning brain anymore. But just simply because of the cellular mechanisms involved, they could find their way there. They put them next to some hippocampal tissue, which really they shouldn't want to have anything to do with, and yet they grew unconstrained in it, so they obviously didn't know where they were going. Yeah, and it was not something they would normally probably come in contact with. And they put them next to some cerebellum tissue, and they went, "Ooh, I don't want this, I don't know what to do," and they turned away. So you sort of get the impression from this that these processes are quite innate, really, the way in which the brain develops, maybe. But we have to be very careful about how we use these words. And, actually, if you've got... if you go to the Johnson and Morton book, which is one of the ones that have been recommended in your reading matter for this unit, you can see that they're basically trying to preserve the word innate for the internal environment. So yes, we can use the word innate in that process because there's no external environment coming into that Petri dish via the thalamus, because it's not connected to an organism. It's purely about what the cells do in a Petri dish, and therefore we can use the word innate. But if you said to me, is being a boy or being a girl innate? I'd say, ooh, I don't really know about that, really. I don't really know about that, because there are all sorts of things that can occur that can influence testosterone production during gestation. And, so I start getting... you have to be quite careful with these things. On the other hand, then, what happens when the whole organism is involved? Well, that's kind of... if a whole organism is interacting with the environment and there is an environmental interaction, then we could call that a type of primal interaction if it's expected. So, for example, if you take little kittens, I'm not sure you'd want to do this, but you patch up one eye. If it's patched up for a couple of months, their vision will never really recover, because that little kitten's eye is expecting to see light. And if light is not there during that period of development, then you're going to have a problem with its functioning later on. So that's an expected species typical sort of response to get. On the other hand, when we can also change, we can also learn in an individual way. So when we learn something, that changes our biology. It has to be sourced somewhere in the brain. And so that type of interaction with the external environment we call learning. So you have to start thinking about these interactions with the environment in a different way. If it's interacting with the internal environment, you can still call it innate. If it's interacting with the external environment, then we can call it a primal species typical interaction, or we can call it learning if it's individual specific to actually us as an individual, so that my experience is going to be different to yours. So is there any interaction that is actually responsible for areal structure? In other words, this mapping of the brain in terms of Brodmann areas. And all these different coloured areas are Brodmann areas, which appear to be self-organising and different, this microstructure. And it's strongly associated with function. As I said, it's always a bit messy, but essentially you can say that Brodmann area 1, 2 and 3 are for sensory information, Brodmann area 4 is for motor. Well, how did the neurons know how to go there and produce that areal structure, that structure of the area? So there's two theories. You could say, well, it's early differentiation into cortical regions according to these internal intrinsic factors. There's no brain activity required in order for that to happen. There hasn't got to be any input into the brain. These are the sorts of things we're talking about with glial cells, yeah? Or you could say the protocortex theory, which is in fact, it does depend on input from the thalamus. Now remember, the thalamus is the main input to the brain. So we're just saying it does need environmental input in order for this areal structure to form. And in fact, there is evidence for both of these things being correct. We've already talked about the Petri dish experiment, but we also see that rodents who haven't got thalamic connections, now if you cut the thalamus, then there's no input coming into the brain, apart from smell. I think they must have got rid of that one as well. Maybe it's different in the rat thalamus, I don't know. but there's no input coming into the brain, and yet they still get something that looks like reasonable cortical organisation. So you could say, well, okay, it must be innate then. It must be purely natural. But then we have all this other evidence that says, no, the way in which the cortex can organise itself actually can be very influenced by the input that's coming in through the thalamus. So we see spontaneous activity in the brain before birth, which appears to be indicating what you'll get in terms of areal structure afterwards. We get later plasticity in terms of the brain. So, for example, somebody who loses their sight, their visual cortex can start being used for something else. So even those primary sensory areas can shift according to what happens later in your life, later experience. And we also know that these gene expressions tend to give rise to graded maps through overlay and different combinations, so that even the gene expressions, even though we may have a limited number of factors or issues involved at a genetic level, the multiplicity in terms of the different range of outcomes that can produce are very many. Some further experiments on this show that reducing the thalamic input to a cortical region reduces its size. The rewiring of the thalamic inputs causes a new target region to take on the properties of the normal target tissue. So if you say, here's my output coming out of the thalamus, I'm going to disconnect it from the visual cortex and put it into the auditory cortex, or you'll start finding that will actually, the neurons here will start taking the properties on that these neurons should have had. So the transplanted cortex takes on the characteristics of the new location. Neural activity appears a critical factor in areal development, so it's not just innate. And the answer is, as with all these things, it's an interaction of genes and the environment, an interaction of nature and nurture. So development is all about an interaction of nature and nurture. The biological structures emerge from a complex interaction between genes and the environment. That's not a new thing. It's just that it's much more accepted now, I think, in the last few decades. It was said by Piaget many years ago. The phenotype, therefore, the outcome is equal to the genotype interacting with the environmental history, if we leave out all these other issues. Now the genotype, of course, and I'm now going to tell you it's not that straightforward. The genotype is equal to the genes, essentially. It's the heritable part. But we're now finding out that you can inherit characteristics without having any change to your DNA, which is quite freaky. When am I going to tell you more about that? In a minute, I think. Our genetic background is also very important for educational outcomes. I thought I ought to put this in because it seems relevant. For example, since you're all educators, amongst UK 9-year-olds, most of the variation we see in UK English, science and maths achievement is genetic. In other words, I think it's something like about 60% or 55% or something, but just over half. Over half of the variability we see between children at GCSE or whatever examinations appears to be explained by their genes. And we have to be very careful about this, because actually that figure is going to be a function of the schooling system. Because if we had schools that were absolutely identical and homes that were absolutely identical, then it would be 100% determined by genes. Do you see what I mean? So what it's not saying is that we don't have to worry about education. Well, that's not very helpful. I don't want to put that on. It's not all about genetics. That's not what that's saying. Okay, it's very important to realise that. But it does mean that genetics is an important issue in terms of understanding how children get on. Also, life is probabilistic. Biology is not destiny, but one theory is that there is a progressive restriction of fate. The idea here is that life, development, or life, is like a little ball bearing at the top of a mountain. And a small perturbation early on, so a little earthquake here, can knock this ball bearing down there, or it can knock it down there. If it knocks it down here, it's never going to come over to here again, because you've got this huge, loads of waves in the way, loads of mountains and ridges in the way to stop it from coming over. Whereas if it gets a little perturbation here, then it might go left or it might go right, and maybe if you give it a bigger perturbation, it might jump over. But essentially, the more you roll down the mountain, the more you're restricted you are in terms of your fate. So it's a progressive restriction of fate, in some respects. Having said that, the brain is plastic, and you learn all through your life, I should say. And learning does change your biology in some amazing ways. So the central dogma is that DNA produces RNA and produces protein, the structures. And you have to be careful here, because I'm going to start using the word epigenetic, but if you come across the word epigenetic, it has two different ways in which it's being applied. The way that Waddington, who produced that landscape, applied it, he just meant all environmental influences on development. Okay? But now, epigenetics, not epigenetics, that's a spelling mistake, epigenetics has come to be applied in an entirely different and much more radical way in terms of how we're understanding biological change. And it basically means all heritable changes that are not caused by changes to the DNA. Yes, that's just what I've said. So do be very careful. There's two different meanings. One example of that is methylation. And it appears during one's lifetime, particular experiences, and we know most about this in terms of stress. For example, with rats who don't receive enough licking and grooming from their mum. Methylation can occur to their DNA. And these methyl molecules, I suppose you could call them molecules, CH3, attach themselves onto the DNA, and they actually stop the DNA from expressing itself. And that brings about change in the organism, but it also means that change can be transmitted, because when the DNA gets transmitted, those methyl molecules appear to be transmitted as well. We don't really understand all about how this happens. But the important thing is that it can produce rapid changes that are almost evolutionary, except they're not changing the DNA, so they're being inherited over generations. And that can be a good thing, because it means that you can adapt to the environment very quickly. So if your mum has had a particularly harsh environment, it can be helpful for a baby rat to already come out kicking, if you like. But in human environments, we like to think that those sorts of experiences are much more limited, at least to one generation, or we want them to be. And therefore it can be a negative thing, because it means that over the next couple of generations, you may end up with offspring that are behaving as if they're in a highly stressed environment, when in fact they're not. And that can be maladaptive, because they're not fitted to the environment. There are different views on how we should think about genetics. So my colleague Robert Plomin will tell you that DNA has a unique causal status in developmental outcomes due to its unidirectional influence. In other words, nothing can change DNA. It's all about DNA driving change, and therefore it's something we should really be, because it's quite determining in that sense, it should be taken very seriously. We have specialist genes that are linked to reading disability, but we also have a large number, we think, of generalist genes that appear to influence outcomes in a number of different subject areas across academic achievement, across cognitive ability. And if we could understand perhaps genetic background more, then we might be able to tailor education to genetic profile. This is one possibility. On the other hand, it could be argued that genetics provides probable, not certain, outcomes. And although microbiologists often assume this DNA to RNA to protein approach, we know that in fact there are causal mechanisms going back in the other direction. Epigenetics is one example of that. The DNA is not actually being changed, but it is being. But the way in which the DNA expresses itself is being inherited. And I would try to turn you on to the idea of neuroconstructivism. Neuroconstructivism rejects this maturational unfolding of the brain, that it's all sort of predetermined, if you like. It's not all about the unfolding of pre-existing information in the genes. It's actually a process of construction between our genes and the environment. Biology is not destiny. That's the most important message, I think, that's coming out of a lot of modern science in this area. So, according to this philosophy, cognitive and neural outcomes emerge from a complex bidirectional interaction between inputs. So, genes, environment, interact with each other, sensory, cortical activity from different regions interact with each other. Evolution is argued to have selected for adaptive outcomes and a strong capacity to learn rather than prior knowledge. So we're not born with a brilliant language skill, but we might be born with a predisposition to acquire one, given appropriate environmental stimulus. It's more plausible to think of domain-relevant mechanisms that become domain-specific because of the experiences we receive, such as in school. So more and more we're thinking about the cortex as quite, not a blank slate, because it has a preparedness to learn in particular ways, but it's certainly quite general in what it can do in many respects. And if you lose your auditory cortex, or you lose the input to your auditory cortex can serve to do other things. It's that general. And all of that means that we need to think about development in the brain in different ways. We can think about things as being probabilistic, that early interactions, early interventions are possibly more important, but that in fact interventions all throughout life are likely to have some effect. We can also consider that the common stimuli, because I said preparedness, and for your brain to have preparedness, something that has to have evolved, then the stimuli that it has to be prepared for have to be very common. So we can assume that the brain might be prepared for faces. It might be prepared to hear voices. Is it prepared to recognize flowers, recognize different animals, as some scientists in evolutionary psychology would have us believe? I don't think so, because we're only going back 100,000 years there, which is not very long on an evolutionary timescale. But those common stimuli, like faces and like voices, such thing, we can expect the brain to be prepared for those. Also, if construction of the brain is influenced by neural inputs from other areas, then atypicality in one area will produce atypicality elsewhere. So if you have a difference in this part of the brain due to maybe a dysfunction in the thalamus, you may expect other differences in the brain as well. Okay, so that's important. So just because you found a difference here doesn't necessarily mean it's causal. And most important, I'd like you to remember this, systems are going to be much more vital. If you're talking about a domain general cortex, then those subcortical bits underneath the cortex, underneath the duvet, okay, things like the amygdala and the thalamus and the midbrain regions, those are likely to be very influential when it comes to how the cortex is getting programmed. And it also means that damage to those systems is going to be much more devastating to development. If you've got only half of one of those, you can expect to have a very different cortex, and a cortex which is really struggling. Whereas if you can actually get rid of half the cortex, so there's amazing stories done by my colleague Helen Imedino Yang of boys who have actually, due to tragic circumstances, have had half of their cortex removed, they've had a hemispherectomy, and yet they go on to experience fairly normal lives in schools, and actually their development and achievement is certainly not at the bottom. They're doing okay. They're not at the top of the class, but they're not doing badly considering half their brain is missing. Okay, so that tells you something about how important some of these systems underneath the cortex are. And it also means that different types of initial atypicality can result in the same outcomes if the same one brain system is affected. So, basically, you could actually have a whole range of different cortical behaviours, but actually they're all arising, so in different people. This person's got different cortical reasons affected than this person, but actually it could arise out of damage to the same system. So that's why it's important to think about neuroconstructivism. This is a famous model that was produced by Uta Frith and John Morton, and it's really emphasising this idea of brain, mind and behaviour, and this is something I really want you to start thinking about. You're here to become psychologists, and as psychologists you're going to be developing models based on behaviour. That's what psychologists do. How good are those models? In psychology, if they explain the behaviour, they're probably good enough. But when we get to cognitive neuroscience, we like to add an additional constraint. Those models don't just have to reflect the behaviour, they also have to reflect what we know about brain function. So then we have this problem of how we should think about the mind, which is what psychologists are doing, thinking about all the time, and the brain. And the best way to think about this is to think about the brain-mind-behaviour model. This is what we have here. Now, when this was first produced, it was really produced in terms of development. And so Uta and John really just put one set of arrows rather unidirectionally going from brain to mind to behaviour. And I've messed about with it a little bit here just to emphasise the fact that actually it goes back the other way as well. And as educators, we're probably more important, we're probably more concerned with the other way, actually. Because if you stop somebody, well, this is how it works in terms of development. If you have a problem with your thalamus, then that is going to influence let's say the amygdala, that's an emotional processing. That's going to influence your ability to think about emotions, and that's going to influence your behaviour. Okay, that's fairly straightforward. But if you're in a school and you don't have access to books, for example, or you're taught poorly, then you will not be able to have the mental concepts of other children. And if you don't practice those mental concepts, they are not going to be influencing your brain, because mental concepts get stored in the brain, and therefore that will affect your brain structure and your brain connectivity and your brain function. You might find it odd that it changes your brain structure, and what I'm talking about there is bits that get smaller and bits that get bigger. But actually that's what happens. You can be, if you're training to juggle, or for example, London taxi drivers, the size of their posterior hippocampus, very important for memory, is actually proportional to the number of years they've been driving their taxi. The number of friends that you have on Facebook is proportional to the regions in your brain responsible for social memory. Okay? It actually changes the size of the different parts of your brain. So we know that it's not just one direction, it's the other direction as well, and that's really important to think about. We've talked about environmental factors here. They put environmental factors on the other side. I don't want you to get too far into this diagram because I've redrawn it. I prefer this diagram. Here we have a brain, mind and behaviour model of a person, exactly the same thing. And cognitive neuroscience is very good, particularly at looking in that direction. They're not quite so good at looking in the other direction, but they're working on it. Okay, it's getting better. But I've drawn another one on the other side, because actually social interaction is very often the basis of education. And they're communicating with each other. And they're using language. And cognitive neuroscience is not good at interpreting language. Okay? It's not good at interpreting meaning. Social sciences are very good at that. So when you're doing your independent educational inquiry, you'll be using qualitative methods, and you'll be using social science approaches to understand how two individuals or more interact with each other, how they express themselves, and you'll be interpreting it using your qualitative methods. But underneath the skin, in the brain, and also in the mind, which is just it's a theoretical concept. You can't touch the mind. You can touch the brain. Underneath the skin, we have these brain-mind behaviour models that cognitive neuroscience is trying to investigate. So all of these research methods are relevant. That's what I'm trying to say. Now that's quite a heavy lecture in a way, because I've introduced some things that I really want you to go away thinking about. I also, sometimes I feel like putting up a slide saying this is not the truth. Because I want you to be thinking about, well, how should I think about these things? I mean, in summary, this is the way we're supposed to think about it. This is the scientific model. But I just want you to know it's not that straightforward, and we have to think about it very carefully. Have we got any questions about that? Yes?
I was reading The Blank Slate by Stephen Pinker.
Right.
There was a bit about epigeneticists and how they were pretty much vilified a while ago, kind of almost done to the fascists for saying that the brain isn't a blank slate. How widely accepted are those kind of views today?
Well, I think what you'll find is it depends where you go. So, you know, I think there are people who are trying to join all this together, and it's possible when you do that to understand that actually it does all begin to make sense. But what we have are silos of people working in particular specialisms. So if you go to somebody who does nothing but genetics and biology, they will still believe that everything is 100% determined by genes. If you go to people who are dealing wholly with social science approaches to learning, they will tell you that it's nothing to do with biology, and these scientists with their white coats have too much authority, and we should forget all about it. And then we have people who are trying to work in between these two things, and that's painful, because there are still plenty of fascists around. Yes, there's still plenty of people who want to. No, it's not the right word to use. But there's still plenty of people who believe, you know, wholeheartedly in a particular dogma. The thing is, I think we have to keep an open mind. That's the most important thing. But you know, even between psychology and neuroscience, there is a friction at the moment, because as neuroscience steps forward, a lot of psychologists are saying, actually, I don't think we need neuroscience. It doesn't tell us anything new, and that is really not true at all. What neuroscience is doing is making it more difficult for psychologists, because if there isn't a neuroscience basis for a model, let's say a model of working memory or whatever, pretty soon that model is going to be out of date and it's going to be challenged. We know that, because it has to make sense at some point in terms of brain function. And that's the important thing. But what you guys have stepped into is I'd say a theatre of debate rather than a theatre of war, but a theatre of huge debate. And it's changing, and that's why slides have to change every year, especially on this unit. You know, in epigenetics, I can remember just a few years ago when there was a sense of, do we have to take this seriously? Oh, yeah, but now, yeah, we very much do. Any other questions? Okay, can I remind the neuroscience and education people that we're having a little trot down to out Bristol. So we're just going to be meeting at the front door of 35 Berkeley Square in 5 minutes. If you have any time to grab a sandwich, that would be great, but we need to be down there for 2. So I'll see you there in 5 minutes. See you all next week. Thank you very much. Thank you. Oh, thank you very much. Thank you. Anybody not signed the register? Anybody not signed it?